Hello everyone, welcome back to the channel for doctors, and today we are going to start the microbiology section. So in microbiology, we are going to study the basic bacteriology. So the basic bacteriology starts with some bacterial structures that are important in understanding the bacterial pathology. So starting with the appendages. Appendages is of two types. It includes the flagellum and the pilus. So flagellum is made up of proteins and it helps in the motility of the bacteria. The pilus is made up of glycoproteins and it helps mediate adherence of bacteria to the cell surface. So the cell surface could be of the host cell or it could be another bacterial cell. If a bacteria holds an adherence with another bacterial cell with the help of pilus, this is known as the sex plus formation, and the sex plus helps in the conjugation of the bacteria. So conjugation is a process where there's a transfer of genetic material from one bacteria to another bacteria, and this is how there is a transfer of important enzymes, transfer of important resistance against antibiotics, and other variety of pathogenesis that we are going to study in our future sections. So there is another specialized structure in the bacteria, that is the spore formation. So spores are basically made up of keratin-like coat and they contain the peptidoglycan and the DNA. So spore is a characteristic feature of the gram-positive bacteria and it helps survival of bacteria in dehydration conditions, in the heat conditions, and when in the chemical environment where there is not a favorable environment for the bacteria to survive. The cell envelope of bacteria is made up of capsule, that is a discrete layer usually made up of polysaccharides, and it helps protect the bacteria against phagocytosis. Similarly, there is a slime layer, that is a loose network of polysaccharides that mediate adherence to surfaces and plays a role in biofilm formation. Then we have outer membrane of bacteria, that is made up of three components. It is the outer leaflet, then we have the embedded proteins and the inner leaflet. So the outer leaflet contains the endotoxin that is specific to the gram-negative bacteria only, and it helps mediate the pathogenesis of bacteria in producing septic shock, and that we are going to study in detail in our in subsequent sections. Then you have the embedded proteins, that is the porins and outer membrane proteins, which help bacteria in transfer of material, and some of the outer membrane proteins are also antigenic. Then you have the inner leaflet that is composed of the phospholipids. The phospholipids are another important membrane structure and it helps in the bacterial cell maintenance. Then you have the periplasm. So periplasm is a space between the cytoplasmic membrane and the outer membrane in gram-negative bacteria. It is made up of peptidoglycan, that is usually a middle region. So the periplasm accumulates components of the exotic gram-negative cells. For example, whatever the structures that are synthesized in the bacterial cell, they are usually transferred outside and they are accumulated within the periplasm. So it contains various hydrolytic enzymes such as beta lactamases. Then you have the cell wall. So cell wall is a peptidoglycan, and basically it's a sugar backbone with peptide side chains that are cross-linked by a transpeptidase. So it is a net-like structure giving rigid support and protecting against the osmotic pressure damage to the bacterial cell. Then the last we have the cytoplasmic membrane, that is just like eukaryotic cells. It is a phospholipid bilayer sac that is embedded with proteins, but the specific proteins for bacteria to survive are the penicillin binding proteins and other enzymes. So lipoic acids that are characteristic feature in the gram-positive bacteria only extend from the membrane to the exterior. So cytoplasmic membrane is also a site of oxidative and transport enzymes. Penicillin binding proteins are involved in the cell wall synthesis, and lipoic acids induce the formation of the DNF and interleukin 1. Here you can see the structure of the bacteria. There is a flagellum, then you have the pillars. This is the outer capsule. So this is the cell wall, and this is cytoplasmic membrane, which is a phosphobilipid layer. And there is wide periplasmic space that contains the beta lactamase enzymes. This is the endotoxins. This is the porins, which help in the transport of material. And So these features are specific to the gram-negative bacteria, which is the enterotoxin and porins and outer membrane, and lipoic acid, which is specific to the gram-positive bacteria. So this is what you need to remember, that is the gram-positive bacteria uniquely contains the lipoic acid, and gram-negative bacteria contains the enterotoxins or the lipopolysaccharide that helps in the disease pathology of the septic shock. So this was all about the bacterial basic structures. In our subsequent sections, we are going to study about the gram stains and the different type of bacteria. How do we organize them into two categories and further their distribution? So if you have any questions, you can ask in the comment section, and we will see you in another video. Thank you.